Thanks everybody for showing up to the seminar today. It is my pleasure to introduce you Dr. Nathan Springer. Dr. Nathan Springer is a professor, let me see if I remember, how can I say, a McKnight endowed professor at the University of Minnesota. Sorry about that. His group uses a combination of classical genetics, molecular genetics, and genomic approaches to study molecular and inheritance variation in maize. He is also the director of the Microbial and Plant Genomics Institute at University of Minnesota, and he's an editor for the PLOS Genetics and Plant Physiology journals. That's it. Please join me in welcoming Dr. Nathan Springer.
Well, thank you very much. It's wonderful to be here. Those of you who know me know I'm not very formal, so if you want to interrupt at any point, please do. I'm happy to talk about these things. I've seen the end, so I don't have to get to the end today. I'm going to talk today kind of about transposons and about epigenetic variation. I will tell you lots of work done by these folks. Sarah is a current postdoc. Jackie is a current graduate student. Ching's a former postdoc. Steve's a former grad student. And Michelle is a graduate student at Jeff Ross Ibarra. But you'll see their pictures again. I have lots of wonderful collaborators, and I'm grateful for having funding so I can do something. OK, so we're going to talk about variation. And I'm going to tell you about variation in corn. You can see all sorts of phenotypic variation here for floral traits, ear traits, plant architecture. You can't really see them from there. Much of this variation is due to genetics, but there are classical examples where epigenetics, two individuals with identical genotype but differences in phenotype, occur as well. And a lot of these have been discovered in maize. So where is this variation coming from in the genome? What are the things that trigger this? So the maize genome has been sequenced for several years and several different times. If you look at this genome, depending on the annotation used, a small proportion of the genome is gene. The majority is transposons. If you look across the genome, so this is taking the 10 chromosomes of maize, there are more transposons near the centromere, but even near the arms of the chromosome, you will see that 50% or more of each megabase is from transposons. So really, like most crop genomes, the genomes are mostly transposon with a couple genes sprinkled in. Now, I've said transposons a couple times. So transposons are actually a large number of different subtypes. Okay? And I'll try to impress upon you a couple times today that thinking of them all the same is probably a mistake. There are a variety of behaviors for different transposons. The two major groups are the class 1 retrotransposons. I'll often refer to these as LTR elements. And the class 2 DNA transposons. So these retrotransposons need an RNA intermediate. They need to be expressed in order to move. The DNA transposons don't need to be expressed. They utilize a DNA intermediate. Okay? A functional protein has to be made to help them move. I will mostly talk about the LTR retrotransposons and the TIR transposons today. Now, if you look at the genome, so this is a very old figure from Jeff Benetson's work. So this gives you a typical idea. So there are 3 genes here in this region. And each of these genes are flanked by all sorts of transposons. In a broader sense, so if we're looking at a megabase of the maize genome, here's a megabase from near the end of a chromosome, here's a megabase in the middle. All this red stuff is transposon, the blue stuff is genes. So these genes are residing in amongst a bunch of transposons. Near the centromere, there's lonely genes and there are no other genes around. Okay? So really we can start thinking about these as what is going on with the neighborhood of these genomes? So as you think about this analogy, I thought a nice way to show you this was to show you my neighborhood. So this is where I live. My house is right here. Some people screwing around in my backyard at a recent lab party. So you think about my house as a gene. I can think about transposons in my neighborhood. So when I first moved in about 10 years ago, ever since then, right back here under these trees, there's an old camper. I have never seen somebody go in or out of it. It's a piece of junk. It just sits there. It doesn't affect me, though. These trees grow pretty nicely. As long as it's not spring or winter, I can't even see it. There are other things, though, that affect the neighborhood in other ways. So fortunately, it's a little ways down the street. But this house here has a much more active effect on the neighborhood. So this is a picture from their yard recently. I won't tell you all sorts of things, but it's not a wonderful presence in the neighborhood. So you think about different genes, and they're living in these neighborhoods, they're residing, they have to deal with their neighbors, which are often transposons. So we can think about ways that transposons affect genes. Often we think about them as insertional mutagens. They jump into a gene, they break the gene. But there are all sorts of ways they can affect genes without directly breaking them. So they can insert into enhancers or repressors and affect their regulation. They could bring with them their own regulatory information, splice sites or promoters, or they could affect the epigenetic regulation or small RNAs. So in each of these cases, they're affecting regulation of genetic information. There are nice examples in plants where this affects the phenotype. So today, I'm going to spend about the first half, or roughly, talking directly about transposons, variation for transposons in the genome, their effects on the transcriptome. And then I'm going to shift to talking about DNA methylation, and you'll see that transposons are woven into that as well. Okay, so this is a slide from about 10 years ago from one of Hugo Duner's papers, and I really like this slide. It shows, in this case, I'm just showing a part of the figure. The yellowish or goldenrod colored things here are the genes, and I'm showing you 3 different haplotypes. So largely the genes are the same, but the transposons that are upstream of a gene can vary radically between different haplotypes. So the neighborhood and the transposons that a gene has to live next to are quite different among Maize haplotypes. Now this was a lot of work. This was done by sequencing back selected from back libraries in these cases. So we know a lot about a couple of loci. We haven't had a good picture of the whole genome and differences in transposons. Just got bit by a bee. All right, so I can keep going. If I swell up, it turns out I'm allergic to bees, so we'll find out. So the first thing I want to talk about is we need a good annotation of transposons. Okay? So originally, the original reference of the Maize genome in many genomes that get annotated for transposons are done by a repeat masker type approach. This is nice at finding fragments of transposons. However, it finds lots of elements and it breaks them into little pieces. So one transposon, sometimes in the annotation, is broken up into 8 or 9 little pieces, especially older elements. Recently, Michelle Stitzer has been doing a structural annotation. So this attempts to find all of the pieces from an element and keep them together. So her annotation has a lot fewer elements, but they're much longer, and they have many more families. Essentially, these 2 approaches, they both find a lot in common. The repeat masker finds a bunch of little fragments that are probably old, decayed pieces. The structural approach finds a lot of low copy elements not found the other way. So she has been doing this annotation, and this helps you to reconstruct these nests of transposons and know that this is just 6 elements across this region. In addition, there are a number of people here in this room and at a bunch of places that started working on getting a good assembly of the W22 genome. So a second Maize variety and getting a good assembly. It is a high quality assembly. I'm not going to go over those numbers. If you look at some of these regions of the genome where we know the transposon structure, largely the assembly captures these. So like in this region, the only thing the W22 assembly misses is a gap in the middle of this one transposon. The rest of them are all sequenced. So we have a genome. Michelle has done an annotation so we can look at the transposon content throughout the genome. And what's interesting is starting to look at content differences or similarities of genome-wide. So if you look at the proportion of the genome, you see that they're virtually identical amounts of the genome that are LTR elements, TIR elements, helitrons, or exons. So genome-wide, the percentages are about the same. However, when you start looking locally, you find lots of differences. So here are 2 different examples. These are genes. The dark blue are exons. The red is a TE that's present in an intron of one haplotype but not the other. There are lots of these. I don't know the exact number, but we trust at least there's probably 1,000 of them. TE polymorphism in genes. When we look at families, so here I'm showing you a couple of representations. So these are TIR or DNA transposons. These are LTR retrotransposons. The things in green are families of transposons found in both B73 and W22. The things in blue are only found in B73, things in red are only in W22. So there's actually a large number of transposon families that are specific to one or the other line. If you look at the families, so I'm just looking at the ones in green now, and we're looking at copy number in B73 versus W22. So in general, copy number is similar, but there are lots of families that have more copies in one or the other genome. Perhaps the most interesting way, so we're working on a genome-wide, essentially a syntenic analysis of TEs to identify which these are syntenic and not. We started for now with the simple cases. A bunch of these transposon families have only one member in both B73 and W22. Okay? Of those families, 60% of them appear to be the same insertion, the same place. 40% of the time when you only have one in both genomes, it's actually on a completely different chromosome. So even though they only have one each, they're completely different insertions. Okay? So there's a very large number. We've just started this analysis of a genome-wide TE diversity. I'll know a lot more in a couple months, I would guess. So I'm going to shift from telling you that they're variable, hopefully you'll believe me that they're variable and we'll have a more detailed answer soon, to talking about what is their impact on the transcriptome. So we'll start with direct effects, that is expression of these transposons themselves. So part of this is, one of the questions you students should often ask yourselves is, why didn't we know this answer already? So with transposons, one of the reasons we don't know is that they're highly repetitive. So it's hard to think about how we map these, how we count expression of transposons. And everybody likes to view things in a browser. I do too. With transposons, we decided that wasn't a good plan. In part because so many of these families are high copies. So with genes, when we map reads, we take unique mapping reads. Some of these unique mapping reads in the genome map uniquely to a transposon. And that transposon can be somewhere between genes or within a gene. Well, you also have all these multi-mapping reads, and perhaps the most interesting transposon expression is multi-mapping. We don't know whether a read came from this element or this element, but we do know that it comes from this yellow family. So we started mapping counts, both unique mapping and multi-mapping to TE families, and getting family expression levels for TEs. So we've done this on a couple of the developmental atlases, one from Justin Wally and one from Scott Stelpflug and Sean Kepler's lab. So we have lots of different tissues. I think we're up to like 96 tissues of B73 now. And there are a couple of naive expectations. So one of them is the transposons are over half of the genome. They'll be over half of the transcriptome. They're clearly not that, but the other expectation is they're all silent. They're not all silent either. I would say in general, what we find is about 5% of the transcriptome is coming from transposons. If you look across all of these different tissues, most of them are right around this 5% mark, about 4 to 6%. Endosperm, particularly late endosperm, is different. It's not, everybody's excited by that. It's just 2 transposon families, and actually probably one of them is an annotation error. So I don't think that this late endosperm is highly significant. There's an unannotated zeon in the middle of that one transposon is captured, and that's what that expression is really coming from. So, yes? Are you defining a family? So families are defined in the annotation process, and it's based on LTR or TIR identity across them. And I think it's a 80% identity rule that's required for defining families. There are some things we've done to make larger metafamilies, like, oh, these 4 families are all closely related, even though they got split. So you can do some collapsing higher up, too. But they basically come out of this annotation that Michelle is doing on the genome, where she's taking all of the TIRs or LTRs and clustering them. Now, one of the interesting things about this is that this could simply be a reflection of noise. There's so many of these in the genome, they're getting reads at random. We looked at genes and TEs. So in blue here are genes. And what we're measuring is tau, which is a measure of tissue specificity of expression. Genes, there are groups of genes that are constitutively expressed in whatever tissue they're on, whatever tissue you look at, they're on. There are also genes that are tissue specific. Transposons are largely tissue specific. You see very few constitutive. In other words, these transposons are really showing dynamic patterns during development. They also show dynamic patterns in response to abiotic stress. So the total amount of the genome that is coming from, or transcriptome coming from transposons goes up a little bit in cold or heat stress seedlings. But if you look at particular families, so here are families that are highly expressed in heat or salt. This family is only turned on in cold stress. Here's a family that's turned on in a whole bunch of stresses. So we're seeing dynamic responses of the expression of these transposons. I don't think they're just coming from noise. Now the other thing I'll mention, and we're really trying to figure out how to think about this, is we actually think there are two modes of expression. There's a mode of expression like this one on the top, where you're seeing reads coming across the whole element. And then there are reads, transposons like this, where you see patches of expression. This one is over the LTR. This one, there's just a patch inside. So this is the kind of expression you would require to actually get movement and functional activation of this retroelement. If you don't get expression of the whole thing, it can't move. These ones, on the other hand, might be something where the genome is tricking the transposon to express a little bit of itself to let it recognize it and silence it versus small RNAs or other things. So we get both complete and patchy expression, and those are probably different classes, and we're going to need to break those apart. We're doing lots of different things with this, so we've been looking at the MOP1 mutants. I'm going to zoom through this because I have other things to get to. So we can count families that are changed in an environment or a genotype. One of the things that's interesting now is that we can compare expression. So here's a case where in the mutant, this transposon that's in green, we see lots of reads coming from it. So this transposon is turned on in the mutant. The gene isn't affected. It's expressed at the same level in the mutant and wild type. Interestingly, this transposon has 4 copies in the genome. All 4 copies are right next to genes. All 4 copies have unique mapping reads that come from the transposon in this mutant. So there's coordinate turning on of the transposon. It didn't care that it was next to a gene or changed the gene at all. Okay? And that leads us into the next topic. So thinking about indirect effects. So before I was talking about the expression of the transposons themselves, let's now talk about what happens to how transposons affect the neighborhood. So I made this slide. I know Pat isn't here, but many of you have heard Pat talk, and he likes to give this analogy of the genome as a manual, and the idea that there's some wiring diagram in the genome that makes a species or makes an organism. So I was thinking about this, and I actually am less interested in that particular diagram, and I'm more interested in how you create variation in that diagram. So I think about how I would vary or what I could do to this. So where does this variation arise? If I tried to vary this, I'm sure mostly the result would be a broken phone, right? So the question is, in nature, how do you make something new and better? How do you get to this new iPhone X, right? So I spent a lot of time thinking about this from the point of expression, okay? So if I have some plant and I put it in unpleasant conditions, it will have a stress response. And often the ability to survive that unpleasant condition is due to a change in expression level, okay? So how do I go from genes that didn't turn on or weren't affected by the stress? What molecular change could happen to a promoter to give you responsiveness? So how do you create novel regulatory variation? I was interested in how often does this arise and what are the mechanisms? How do you create expression novelty? So former graduate student Mandy Waters and a visiting scientist Irina Makarevich started this project that was really designed to look at how often do I get expression novelty. So we profiled expression in seedlings from control, cold, and heat stress, 4 parents and 4 hybrids. You can do lots of analyses. You find lots of differentially expressed genes. We're interested in these types of genes here. They turn on in one parent. They don't turn on in the other. They kind of turn on the hybrid. Okay? So here's an example of 2 of those genes. So these genes are expressed in B73 in both control and stress at about the same level. So the stress does nothing to them. In Missouri 17, the gene is significantly upregulated in the stress condition. And looking at the hybrid helped us decide whether this was due to the promoter itself or some transregulatory variation. So here, what you can see is when the stress happens, the gene expression level goes up, and that gain is almost entirely due to the black allele, the Missouri 17 allele. So only one allele can respond to stress. For this other gene, both alleles respond to stress. So we can use the hybrid and the allelic patterns to decide whether it's both alleles or a single allele, so whether it's a trans effect or a cis effect. And we're interested in these cis effects like this because I want to know what could happen at a promoter to make one allele responsive and the other one wouldn't be. For various reasons, you can't look at a lot of genes this way. You need SNPs, you need a moderate expression. There are other statistical problems here. So we get down to a somewhat small set of genes that we can study. Of those genes, half to 3/4 of them are cis regulatory variants. A small set are trans, and some we couldn't classify. But in total, we have several hundred examples of cis regulatory variation for how well an allele responds to an environmental condition. We're currently going through these and thinking about what happens to local SNPs. Are there allele structures, or what alleles are linked to responsiveness or non-responsiveness? But I will tell you that overall we're finding probably about 3/4 of them are due to this less interesting case that is likely a loss of responsiveness. So the reason one allele doesn't respond to a stress is that some cis regulatory element got broken or deleted. That's, once again, that's dropping your phone, it's breaking something, that's not creating novelty, okay? There are a small number of cases, though, where it looks like it's a gain of expression. This is based on looking at duplicated genes or other species and whether orthologous genes respond similarly there. And in those cases, we like this idea that maybe it's transposon insertions that are bringing with them information. And I'm going to show you why I like this idea with the next couple slides. So this isn't our idea. Barbara McClintock proposed this many years ago, this idea that transposons could act as controlling elements and would actually affect neighboring genes. In Arabidopsis, there's a really nice example of this onsen retrotransposon that is responsive to heat, and genes that it lands near become responsive to heat as well. There are similar examples in tobacco and rice where this might happen. But in all these cases, they found single elements when we're asking the question. One of the nice things is Maize has so many retrotransposons, it's easier to do an analysis. So Irina did this work, and she took all of these different families of transposons and asked, if I take responsive genes to a stress, are particular types of transposons enriched near them? We had several hundred families of transposons that have at least 10 insertions near genes to look at. So the most extreme example is a family that was called NIBA. It has a new name, and I can't remember it because it's a bunch of numbers. This family, there are about 550 elements near genes. 480 of the genes near this element respond to cold stress. We would only have expected about 30 of those. So this is a mass enrichment. Almost over 80% of the time when this element lands near you, the gene becomes responsive to cold stress. We did this kind of analysis genome-wide. So we have 4 different stresses, and these are different transposon families. And if there's any color here, we're saying there's an enrichment for the chance that a nearby gene responds to that stress. So you can find some transposons that are enriched for response of multiple stresses. Others are responding to a single stress. So there's about 20 families out of several hundred we looked at. So this is not all transposons doing this. It's a subset of them. In any one stress, there's only about 4 to 9 families that are significant. But those 4 to 9 transposon families account for somewhere between 5 and 20% of the genes that respond to a stress. So in other words, if you did a Go analysis and you had 100 genes that responded, it could be that of those 100 genes, 25 of them are just because some transposon family is near them, not because they are particularly responding to the stress, or the gene itself was functional for that stress. So once again, I was telling you about novelty. And so to look at novelty, we wanted to look at variation. So this was done with Michelle and Jeff. And what we were looking for were cases like this, where we have a transposon insertion in one inbred but not the other, and is there a responsiveness change? So we found some of these polymorphic insertions for families that were associated. And what I'm showing you here is for 20 or 25 genotypes, none of these haplotypes have the insertion, or sorry, all of these have the insertion, none of these have the insertion, same thing here. So if the transposon isn't there, you're never getting the response to this environment. If the transposon is there, you often have it, although in some cases we still don't get response, and we're working on why that is, okay? But the idea here is that as transposons are hopping or varying among haplotypes, they carry with them enhancers, probably for their own expression, but those local enhancers can also affect the expression of nearby gene and provide a way to give expression novelty and rewire new circuits for environmental response.
So how far upstream is that transposon?
Yeah, so let me just go back a couple of this one. So we took each of these families and we found them. I think our discovery criteria required it to be within 5 kb, and we were only, this enrichment statistic was a 5 kb window. If you take every one, you say, what's the enrichment if we take that window to different? Some of them really only work at a couple kb, and after that you get no signal. Others seem to have a signal all the way up to 10 kb. So I think different enhancers that are carried might have different ability, the distance at which they could work varies. We don't see any evidence that transcripts are starting in the transposon and coming out. That's why I call it an enhancer rather than, so we can't see any reads mapping to the region in between the transposon and the gene, or it seems that it's more of an enhancer. The other reason we think that is it doesn't matter which orientation the transposon is in relative to the gene, it still works at the same distance. Okay, so the things I've told you so far. Transposons are highly variable among haplotypes at the genomic and local scales. They're not entirely silent, and they're actually quite dynamic in their expression. I didn't show you the data on this. Certain tissues, so pollen, reproductive tissues, are the most diverse in terms of what happens to their transposon family expression. I also think that they're important in creating novel expression patterns, creating a response. Okay? It's one of the ways you can move enhancers and regulatory elements throughout the genome. So I'm going to shift now to this second part to talking about DNA methylation and epigenetics. Okay? So I used this word epigenetics earlier. Different people use this word to mean different things. When I use this word, I'm telling you about heritable information that's not solely due to DNA sequence. Now, this could be mitotic memory, so there are cases of things people call epigenetic because early in development a choice is made about expression and it is remembered mitotically throughout the rest of development. In other cases, we're talking about meiotic or transgenerational information. So activity or silencing is remembered even in offspring. Okay? When we talk about epigenetics, many people's minds immediately think about chromatin modifications, things like DNA methylation, histone modifications. And these certainly are present and are a mechanism for most of the examples of epigenetics we know of. However, I would argue that often you can get these changes and not have them be epigenetic as well. So many chromatin modifications are not stable at all. They're very transient and are forgotten quickly, or are transient during a cell cycle even. In other cases, genetic information programs exactly where they should be. So they're not independent of genetic information, they're just a reflection of it. So separating the genetic and epigenetic components of these modifications is important. And we're going to talk about that for DNA methylation. Now, this is an agronomy seminar, and you're asking yourself probably if this really is an agronomy seminar, since I haven't talked much about plants or breeding. But I will talk some about that. So, historically, we've done selection, and it's been based on phenotype. And if you're selecting based on phenotype, you really don't care what the underlying mechanism or molecular difference is. You should capture the phenotypes provided based on their level of heritability. Okay? Genomic selection is a little bit different in that we are now training models based on SNPs and traits in a training population and applying it to larger populations. We're only taking information from SNPs and trying to use that to capture information about the phenotype. This mostly works and works quite well. However, people always want to improve it and make it work better. And one of the questions that has nagged several people is, if there's lots of information in epigenotype that's not captured in SNPs, will we be limited by focusing on SNPs? So in other words, if we had a DNA methylation pattern for the genome, how much additional information would that give us? How much more predictive value would we have for the phenotype? So that's one of the reasons we think about studying a methylome. One of the other reasons that I'm going to talk less about today is the idea that a methylome could help us find or predict functional elements in a genome. So how do we find the regions of functional importance? Okay, so I'm going to start by talking about just the pattern of DNA methylation in a genome, where this is in crop genomes and where it comes from. Now, DNA methylation, we probably shouldn't just use that term. DNA methylation is actually a collection of mechanisms. So it's found at CG sites, CHG, where H is any base other than a G, or CHH. These are done by different enzymes, and they each have different mechanisms of maintenance. So both CG and CHG are very faithfully maintained with self-reinforcing loops. So after DNA replication, these marks are stably maintained. That matters because if you can't maintain the mark after replication, it can't be heritable. CHH also has a maintenance pathway. It's much more complex. It requires a large host of factors. Okay? The location of methylation matters. Methylation found in gene bodies is not important for the silencing of those genes. It doesn't seem to have a direct effect on the regulation. Methylation over promoters, though, often is associated with reduced expression of a gene. So what I'm showing you here is the profile over genes. So if a gene starts right here and it ends right here, I'm showing you the methylation in the different contexts with different colors. And I have both Maize and Arabidopsis here. So Maize, outside of the genes, it's very highly methylated. But you get this great big dip. So genes essentially are a valley in the DNA methylation pattern. In contrast, if you look at transposons, they represent a peak. So methylation goes way up at transposons. There are some interesting differences. In Maize, there's kind of these broad shoulders. In Arabidopsis, it's very specific. So Arabidopsis is very good at very carefully finding the edges of transposons and changing chromatin there. Maize is a little messier, so the idea of neighborhood effects we'll come back to later. This is a different view of an Arabidopsis region, a Maize region, just to illustrate these differences. So I'm only showing you two of the types of methylation here. You notice that their methylation of these two types really agrees with each other here. In Maize, the black and red are not in very strong agreement. Not only that, this mark here, the red mark, is often thought of as a good marker for heterochromatin. Essentially, the whole Maize genome is heterochromatin. There are these little holes that are over top of genes. Other than that, it's marked. In contrast, Arabidopsis, there are just little patches of this. There are lots of differences in Maize and Arabidopsis. The most critical one is hard to show in a graph, and that is how we can learn about function of these. So in Arabidopsis, we have mutants that can knock out any of these types of methylation. You can get to a plant that has near 0% methylation. In Maize, since I started graduate school about 20 years ago, I've been trying to get good knockouts of methylation. I have never gotten a good knockout. We have lots of reverse genetics alleles. The knockouts of a little bit don't matter much. If I knock methylation out about 20%, that's the best I can do. I get a plant that looks like that. And so for those of you that look at corn, that is not a good looking corn plant. Okay? I think anything more, any other double mutants we've done tend to be inviable if we predict they would affect methylation greater than that. This is also true in rice and tomato, so it seems that the ability to tolerate a really strong reduction in methylation is pretty limited, which limits some of what we can do in terms of understanding its real functional role. Okay, so this epigenetic information, one of the important questions is what triggers variation in this? How stable is this? People like this idea. Thomas and I were talking for a while about this earlier. Like, could you create epigenetic variation as a new source of variation? Will environments trigger this? So we're going to talk about how you get these regions of variable methylation. Okay? So at the local level, you're thinking about going from a gene that's active and expressed to something that's methylated and silenced. There are natural sources, so this can happen spontaneously. A spontaneous back and forth would actually be an epigenetic shift if it's maintained and remembered. However, it can also occur due to a genetic change. There's some evidence wide crosses or polyploidy can also perturb patterns. You can also have induced sources. There are mutations, like I mentioned to you, this has been done in Arabidopsis. It's harder to do in crops. Chemical treatments, tissue culture has actually been shown to affect methylation at some regions. Hopefully in the future we can do some epigenome editing. There's also been suggestions that environmental influences could affect methylation or development. I'll talk mostly about these spontaneous ones. These two up here, we're going to start with these. So first of all, development. People think development and methylation will change during development. I'm going to tell you very quickly, it really doesn't change very much. We'll ignore this, it's more colorful, but this is a better illustration from some recent data. In blue, I'm showing you how many differences in methylomes we see between two genotypes of maize. And in red, I'm showing you how many differences in methylomes we see comparing roots and shoots. So it's about 500-fold more variation between genotypes and between the two tissues. And I'm not even convinced that these are real and that these aren't noise. Okay? So between two tissues, if those tissues are not endosperm or reproductive, you see almost no change. Okay? If you take environmental stresses, so we've done some experiments where we took seedlings to near death, and then we grew them and we looked at later stages and asked, does the methylome change? We've also passed things through tissue culture. You can't see this very well. So this is just finding any region of the genome that changed in at least one sample. And just based on the clustering, what I'll tell you is that all these symbols here tell you what environment it went through. So this clump here all went through tissue culture. They have some common differences. Plants that went through heat, cold, UV, they have just as much differences among each other as control plants do among each other, and they don't have any evidence for higher rate or targeted changes. Okay? The source we see that really affects methylation the most is natural variation. So regions of the genome where one inbred has a different pattern than another. I can kind of illustrate this with this big clustering here. So this is about 90 different samples we've profiled DNA methylation for various experiments. I'm showing you only about 4,000 regions of the genome. I can't read the labels up top either, but I'll just tell you a couple things. So this region here, these are all B73 samples. They're from different tissues, different seed sources. Some of them were 15 generations removed from each other. B73 pretty much looks like B73 no matter where you get it from in the plant. Okay? These are tissue culture samples. They're not this different because of tissue culture. They're this different because they all came out of A188, which is a different inbred line. Okay? So most of our variation here is driven by genetic differences in the genotypes, not due to developmental change or environmental change. In any one genotype, so I have to be careful in telling you how much variation there is. There's a lot of differences, tens of thousands of differentially methylated regions between two genotypes. As a percentage, though, of the genome we can look at, CHH varies in about 0.1% of the genome, and about 0.5 to 0.8% of the genome varies in CG or CHG. So in other words, over 99% of the genome isn't changing. That 1% that is, it's kind of like SNPs. Most of the genome is similar. 1% might be SNPs. Now, some of the things that are really important are how stable is this and how is it inherited? So when we have variation, one of the things that we asked, and this experiment actually is really important for thinking about how this would work relative to genomic selection. We're not doing a genome-wide association here. We're doing a local association scan. So we take a differentially methylated region somewhere in the genome, and we take all of the SNPs nearby. We say, are SNPs near this region associated with its methylation level? In other words, could I have inferred methylation just by looking at nearby SNPs? We find is about half of the differentially methylated regions are significantly associated with nearby SNPs. And if we go to new genotypes, if I had their SNP profiles, I can predict at about 80% accuracy the methylation levels for those half of the DMRs. The other half are not tagged by SNPs. So in terms of this idea of genomic selection and SNPs capturing the heritable information, we're finding there's a substantial amount of heritable methylation change that is not captured by nearby SNPs. And this is somewhat similar if we look at a biparental population here. This column here are things that are stably inherited at the local level. These are some that are harder to predict or less certain about. Now, when we think about this question of how they're captured, a lot of it comes down to what we think of the mechanisms that drove these. So these differentially methylated regions, it's methylation, so a lot of times we want to just call it epigenetic. But really, we can have pure epialleles, so a shift from an expressed unmethylated form to a methylated form, that's epigenetic. This can also happen. A transposon jumps in and it spreads methylation to nearby regions, and that triggers the silencing. So in that case, you've got a chromatin change, but it's probably caused by a genetic change. I have one of those wishy-washy answers. I'll tell you both happens. Okay? So we think that there are these pure epialleles. If we look at regions of the genome that are identical by descent, we see about half as many changes in methylation in those regions as we do elsewhere. So even without sequence change, you can still have methylation changes, but they're not as common. We can also find that there are cases where if I know that there's a presence or absence of a transposon, it is associated with the level of methylation nearby. So if I only looked at this region, I would call this a methylation change, and that's interesting. But really, this is predictable by the content nearby. Okay? So that second case is due to these a subset of transposons. And I just want to show you this idea that, once again, not all transposons are the same. Okay? So what we have here are basically 140 different families of transposons. So family 1 through 140. And we're taking the edge of the transposon out 4 kb into the genome and asking, is methylation levels higher than average or not? For these third of the families, they spread silencing marks to nearby regions. These 2/3 of families don't seem to. I still, if you gave me a rice genome now, I couldn't tell you which ones would be which. I have found nothing that predicts which of these 2 families they'll fall into. There's one functional characteristic I'll show you in a second, but we basically have these 2 subtypes, ones that spread heterochromatin, ones that don't. It's not due to the difference in the methylation level in the element. So if I take these top group, they're shown here in black, these ones are shown in red. The methylation level inside of elements is the same, it's just how fast it drops off once you get outside of the elements. Okay? The elements that actually have spreading have no small RNAs. These ones actually do have small RNAs, which was kind of the opposite of what we thought at first. One of the things though that matters here is, so the ones that spread are shown in blue and red. There's a subtle reason for having 2 colors. When those are near genes, so within 500 base pairs, those genes tend to be much lower expressed than if one of those non-spreading elements is nearby or no TE is nearby. So when these are there, they tend to have this effect of that neighborhood tends to have a suppressed or silent state. Now, one of the questions that I really need to answer, and you probably don't think it's all that important, but I'll try to convince you this question's really important, is the exact stability of methylation. So genetic changes rarely change. Maybe 1 in a couple million generations. Gene expression changes all the time. It's very unstable. Within the same cell, you can change the environment and change expression levels. So we often monitor gene expression or SNPs. These are 2 of the things we would measure about a sample. In terms of predictability, though, we use SNPs because we know that they're going to be stable over a long period of time. Where we place methylation in this range and chromatin things really matters in terms of how much predictive value they have, whether they're an effect of something or a cause, and they can be used for information. As near as we can tell, methylation probably falls into this region somewhere in 1 in hundreds to 1 in tens of thousands of generations of stability. Now, exactly where it falls matters. So if you think about making a recombinant inbred line population, if you have 300 individuals, go through 7 generations, the number of meioses you've gone through, you're getting several thousand. Okay? So your disconnect rate, similarly if you do GWAS and you're trying to think how often has methylation changed relative to a SNP, if it's 1 in a couple hundred, you might have enough stability to map in a biparental, but in a GWAS you will lose your signal because of the number of generations you've gone through. So a lot of work right now is going into figuring this out, and unfortunately it's probably not going to be a single answer. Those pure epigenetic alleles might have a very different stability rate than the ones triggered by a transposon. And so sometimes I think the broad range we're working in now is due to the fact that there are multiple causes for these. And until we separate their causes, it's going to be very hard to calculate a stability rate and know how to treat them as additional information that is or is not captured by SNPs. And I'll point out that histone modifications and chromatin accessibility are probably much lower in this range. They're much more variable and less likely to capture heritable information. I also do like to point out that I probably am working on the wrong species. If I want a species where I think epigenetics really, really matters, it's probably things that are clonally propagated, because your ability to trigger changes and have them affect things is greater. In Maize or something like Maize, if we get an unusual epigenetic state and it screws things up, we throw away that plant and we're done. Okay. So the last topic I want to tell you about briefly, and I'm almost done, I promise. I have like 3 slides left. So why does it matter? Do we care that methylation is there? What other information is in the genome? We've done this a couple ways. So one way we've done this is we start from all those regions of the genome that vary in methylation between different lines. We say how often is the gene nearby differentially expressed in an associated way. So there are 2,375 differentially methylated regions near genes. In 277 cases, the nearby gene had a significant negative correlation. And often they were like this. If it was highly methylated, the gene was off. If its methylation went low, the gene was on. Okay? I don't know what to do with this number. It's more than a handful. It's not thousands. So, in a population like this, a couple hundred genes that are exhibiting on-off expression is interesting, but certainly there are more genes than that doing this. And I can show you that better with this next analysis. So here what we did is we didn't start from methylation, we started from expression. You take all genes and you split them into non-differentially expressed, They're different, but less than twofold different, all the way to on-off genes. Then we ask, for each type of gene, how often is the promoter differentially methylated? What you'll see is that the only time when differential methylation starts to become enriched is when you get to genes that are greater than tenfold or on-off. Of on-off genes, things that are on in one genotype and off in the other, now about 20% of those or so have differential methylation. So if you want to look for the effects of methylation, it's not the quantitative changes in expression, it's the on-offs really. Okay? And in that case, those are probably gene expression changes that might be more relevant for phenotype anyway. Now, the other place this matters is beyond natural variation and from the idea that there's cryptic information in genomes. There's information that's not being used. And we've gone through the methylome and looked for that. In our lab, we estimated that there are about almost 4,000 silent genes in the B73 genome that were silenced by methylation. About half of those, not quite half, seem to have natural variation already. That is, a breeder could have captured the active form of that in some other line. The other set of these, though, were silent in all 50 lines we've looked at so far. Okay? Bob Schmitz also did a similar analysis. He predicted about 5,000 cryptic genes in the B73 genome. So there's this idea that epigenetics is reducing the amount of available information that we're using in the genome. So I'll summarize here that there's lots of differences from methylation, primarily in genetically diverse lines. There's a combination of genetic and epigenetic variation. So some of these are captured by nearby SNPs, others aren't. And some of this is associated with altered gene expression and potentially cryptic information. And I think I've showed pictures of people or named collaborators that went. So I'll stop there and take any questions. Yeah.
Very nice. Try to imagine reading implications. So here's a hypothetical experiment. You take two double haploid parents, and you get 10,000 progeny. So it should be identical haploids. But I think both parts of the talk are saying that there are novel events. So what's your gut feeling about what portion of those progeny is going to be different?
So, you know, one thing I want to be careful about with this answer. So often when we look for things that are different, we use statistics. And statistics requires repeatable behavior. So I'm often worried that I underestimate the unstable behavior of the system because things that aren't reproducible in one sample versus the other, you don't tend to keep, right? So there is a chance that there's a subset of information, and we do see a small set of regions in the genome that if I take 10 different B73 plants, even siblings, they'll be kind of random what the methylation level is. So there are some regions of the genome that are very unstable. And it doesn't matter if you take siblings or progeny off of doubled haploids. You'd expect variation for those. Most of the stuff that we found that is reproducible, though, it doesn't matter if I shift between generations. The rate of change is pretty low. And I often worry at the range. So you tell me I get 1,000 DHs. If one of them is different, I'm often going to be suspicious of my technology rather than the biology, right? Did something technically happen as a measurement error rather than a 1 in 1,000 rate? So I think there will be some variation. There's some, but most of the ones we see such low rate of change for the things that are at least partially stable. I don't think there'd be a ton of changes. I could be wrong. I'd be happy to be proven wrong, but that's kind of my guess. Yeah.
Very nice talk. I read some publications about how methylation level was remembered or memorized in the cell going through the meiosis. Can you update us on that front?
So, I mean, there's kind of two ways. Locally, so it depends on the type of methylation. CG, actually the enzyme that does it, physically hangs out with the replication machinery, and it comes right along at the end of the replication fork. And hemimethylated sites. So if there's a methyl group in one CG, the other strand will be CG going the other way. That's the substrate it loves, and it's very efficient at methylating that. That's easy to remember. CHG has a slightly more complicated mechanism. Those ones tend to be locally remembered very well. CHH has an enzyme that's not very processive, and it's remembered less well. But there's also some data in both maize and Arabidopsis that there's some ability to sense and maintain methylation levels. So the demethylase enzyme in Arabidopsis and in maize actually requires methylation in its own promoter. So if it expresses too high and it wipes clean the methylation in its own promoter, it turns off. And if it's off for too long and you get a lot of methylation there, then it will turn back on. So there's a people call it a rheostat for maintaining genome-wide levels. So there might be some ability of a cell to sense genome-wide levels and balance targeted demethylation activities from methylation. But the methylation, which has been better studied, really does have these self-reinforcing loops. So locally, it's remembered. Genome-wide, I don't know that it's as sensitive other than through this demethylation pathway. Yeah?
Kind of follow that question, like, when you look at genome-wide, the methylation difference, you can see that the expression level or like natural relation between the T1 gene itself, like how then natural relation or like expression difference, while in the line or like in the minor difference.
So we don't see large differences in the expression of the machinery. So there are developmental variation in those things, and largely cells or tissues that are actively dividing express high levels of these things. If they're done dividing, they tend to be much lower. Between different inbred lines, there are lots of regions of the genome that vary in methylation, but the average methylation level of these genotypes is about the same. So if you look at the CG context, they're at about 70 to 75% in pretty much every line you look at. Okay, so it's not that one line has less methylation than the other, it's that it's a composite of regions. So there might be 100 regions that are methylated only this line, and a different 100 only methylated in this one, and the average becomes about the same. Yes?
I was looking at the distances in your spreaders versus your non-spreaders.
Yeah,
And in your spreaders, the distances are about a kb or two.
yeah.
So what about these transposonal elements that may have 5 kb extreme and impact on the gene?
So,
What does that have?
yeah. Okay, so there's kind of two separate things going on here that might be opposite. So some of my things that are having an effect on the expression of a gene, those are often positive effects. I haven't looked for that, the responsiveness as a negative effect the same way, right? Actually, when we looked at things that get downregulated and are there that get downregulated in response to an environment, are there transposon families that do that? We didn't find any of those. All of those things I told you about responsiveness were upregulation. So transposons can carry different kinds of information. In that story, I'm telling you about they carry an enhancer. When that transposon lands, that gives it a condition-specific enhancement. This would be more of a silencing activity, right? And so the way an enhancer works is by recruiting transcriptional machinery and doing, that can have a certain size effect, right? And how the chromatin looping or other things happen would be one thing. In this case, it seems to be a more processive thing. And we actually think what's going on here is that over time, so a lot of this information is correlated with histone modifications, and nucleosomes will shift before and after replication. So the problem is if you shift out just for a cell cycle or two, and the histone modification is out a little too far, it recruits the methylation, the DNA methylation. Now that's tagged on there. So like it's this kind of this process of like every now and then even if you're mostly accurate, but every now and then you shift out a little bit, you just keep shifting. It's kind of like telomeres forever shortening. It's the opposite of that, right? So this is a silencing activity coming from the spreading of chromatin. The other is more of an enhancement coming from an enhancer that's somewhere in that element. So that's why I think distance has changed. Yeah?
How would maize without transposons?
I know. Several people have asked that question. What would this genome look like if it had no transposons? I've used the analogy several times that you can have all these different analogies for transposons. People think of them as a host parasite, and there's these nasty parasites you want to get rid of. Probably not entirely true, although there's use in that. You can think of them as the pile of junk. There's just this extra junk, and it means nothing. The analogy I like is how we've changed to thinking about microbes, right? So it used to be all microbes are bad. Now with like the microbiome projects, we think, well, a lot of them have no point whatsoever. They're just around. Some of them are really bad. They still make us sick. And some of them are really good for us. And I think you could think of transposons the same way. Probably you could delete 90% of them and have a corn plant that looked like a corn plant. There might be a couple % of them, if you deleted, corn might be better, and there might be a couple of them, if you deleted, corn might be worse. It's going to be really interesting with CRISPR and other things. Could you go in and make cuts at the ends of a bunch of these and wipe out whole families at a time? Maybe someday we'll be able to answer that question. So now I'm just waving my hands, I'm speculating. But.
There was answering or asking about the amount of transposons. I mean, you showed that 2 lines are very similar, like 90% or so.
Yep.
Amounts. Is there some mechanism to select for or against the amount of transposons?
There's a lot of family-specific variation among lines, as near as people can tell. But there's massive variation in genome size. Much of it tracks back to the knobs, which are not actually transposons, but are these large tandem repeat stretches. That seems to drive more genome variation. There's some hints that maybe that is a selected trait or impacts phenotype. I don't know of copy number or massive shifts in % of the genome that's TE. So.
Maize seems like a unique crop in the amount of mutation that it can carry around. So I wonder, is maize, does that, if you compare maize to other species, Arabidopsis has a lot less TEs, but what about other species in general? And I guess I really wonder if there's a difference between outcrossing and inbreeding species, as outcrossing species tend to carry more mutation.
So I don't think it's entirely that simple of just a cross structure. I mean, even in Arabidopsis, there are some very close relatives. Arapis alpina has a genome that's like 6 times the Arabidopsis size, all due to proliferation of some transposons. A lot of people think Arabidopsis is where it is because recently it's had a very high deletion rate and it's shrunk its genome, not that it never had transposons to begin with. Things like barley have even higher density of transposons probably than maize. The annotation doesn't say that, but the genome size says that's probably true, right? It's a bigger genome with about the same number of genes. It's not necessarily from a duplication. It's probably transposon content. So maize is unusual because there's been a lot of money that went into it. We have a sequence. So in terms of, if you drew a graph of how much we know about the genome versus the size of the genome, maize is kind of an outlier because of the resources that have gone into studying maize. I think we just don't know about a lot of these other big genome species because the resources haven't been there and they're really painful to work with from a sequencing standpoint.
Have you propagated your corn on a plant that has the defective methyltransferase? The issue is, whether or not you see what kind of variation you see in subsequent generations.
I have attempted to. It's pretty rough to get. We've gotten a couple seeds, never more than like 3 per plant, and so it's hard to really then look at variation in offspring. They don't shed well, even if we try to. And that's a double mutation. One mutation is in the CHH pathway. One partially knocks out CHG. And we've made double mutants like to knock out all of CHG, and those we just never even get a viable seed. We think the seed aborts pretty early.
So Arabidopsis, you start getting a whole bunch of variation.
You can eventually, yes. Like the DDM1 in multiple generations, it will throw things off. Even so, the MOP1 mutation maize, you will eventually get you get semi-stable phenotypic variants that get thrown off of that. So some of this probably is, a lot of that is probably there's redundancy in these systems. So these 3 types of methylation give redundant silencing. Losing any one of them just puts you on the edge of now it throws off variants because you've lost part of the silencing information. You haven't lost all of it yet. Yes, sorry. Hey, man.
And they saw that in Palmyra California it was always turned off, regardless of the highways of the 9th edition. Do you have any idea what's going on in the neighborhood there?
I don't. I saw that, and I didn't. I saw the overall finding, and I hadn't looked at the details enough yet to think about why it was that why one genome should have just a preference for doing that. I mean, one thought people have had is that if you really want, so one of the cool ideas of this idea of transposons giving you novel expression responses. That there's actually some, like if you grow them under a stress response where the transposon itself is expressed, it can hop and create new genes that become responsive to that same stress. So there's this theory out there that a prolonged growth in the same kind of stress could create more of that transposon movement that actually creates activation patterns in response to that stress, and that could happen. I don't know of any good natural evidence for that yet. I mean, this could end up being bad if they map back to a family, but that would be the idea, is that things that in a prolonged stress would keep, that basically a sample turning on new genes in that stress is the transposon that responds to it, jumps more and lands more and gives enhancer activity to more genes.
All right, let's thank